Hey everyone, I'm Paras Chopra, and today I'm with Dr. Michael Levin, who's a distinguished professor in the biology department at Tufts and serves as director of the Allen Discovery Center. He holds a PhD in biology from the Harvard University. At Tufts, his research group is interested in figuring out how our bodies know what to become. We all start as a collection of undifferentiated cells in an embryo. How does that ball of cells ultimately gives rise to the precise shape that our bodies have? Surely the answer is not entirely DNA, because all cells in our embryo share the same DNA. Something else must be telling our cells what shape to adapt and when to stop multiplying. Dr. Levin believes that what guides our body plans is bioelectrical communication between different units. Our bodies take shape the way they have because each of our subunits, cells, tissues, organs, collectively decides it to be that way. Dr. Levin is doing biology on the cutting edge, as you'll see quite literally sometimes. I'm excited to learn from him what all are we missing in the mainstream account of biology. Welcome, Dr. Levin.
Thank you so much. I'm happy to be here. Thank you.
I was reading your biography and your sort of career history, and it seems you were a software engineer before you became interested in biology. So how did that happen?
Well, I was interested in biology from a very early age, in that I used to play with insects and watch plants and things like this. But I was always interested primarily in the information processing. I wanted to know, how do these bodies get to be here, and how do their minds work, and how is it possible that some arrangements of matter, meaning living things, are able to have memories and preferences and goals and the ability to process information towards some sort of future activity and so on. So my first real love was biology, but I was always interested in computer science as a way to develop these things from scratch, right? So the idea is that if you can program, that means you're manipulating information from the bottom up. You're defining the ways in which it is processed, and therefore you can test various ways that it might work. So I always was into artificial intelligence, building autonomous things from scratch. And so I learned to program. I was doing some software engineering. I briefly had a software company. I got a degree in computer science, but it became pretty clear at that point that it was really important to understand the biology a lot better to create better devices. And so then I went for biology degrees.
Right. So it's fantastic to sort of listen to this as you migrated from being interested in information processing to biology, and traditionally information processing in biology has been associated with brain, but you chose a different sort of path, which is entire bodies. How did you stumble upon that specific path and not like studying the brain? And of course there are parallels, but your research is much more aligned to the entire bodies and morphogenesis than the just neurochemistry or our brains.
Yeah, well, I was thinking about, certainly neuroscience is fascinating, and of course brains are very interesting, and lots of people work on understanding brains. But two types of thinking led me to this direction. One is that if you ask where brains come from, right? So clearly brains didn't just appear from nowhere. Evolutionarily, basically, neural cells are an elaboration of cells that were here long before brains appeared. And in fact, even bacterial biofilms, and certainly many of the cells in your body do most of the same things that neurons do. They have most of the same machinery, but at a slower time scale, they do some of the same things. So if you think about through evolution how brains got here and what might be the more primitive, older versions of the amazing capabilities that our brains and other animals' brains have, you're inevitably pushed to thinking about what these kinds of capabilities look like before there were brains. What do other cells do when they use the same mechanisms that brains use, and the same kinds of signaling and computation and so on. So that was kind of going at it from the neuroscience path backwards. On the other hand, if you think about embryogenesis, and in particular, not simply the fact that a particular egg can go through the exact same steps and give rise to the same organism every single time. Not just that sort of feed forward emergent process, but actually regulative development, where you can divide an embryo in half, for example, and each half will, if you do it early enough, will give rise to a complete embryo, or regeneration, where these things adapt to novel circumstances and remodel their body structure, right? If you think about that, that is a very computational process. That's a very kind of plastic type of activity that really forces you to ask, what types of computations might there be for the system to really detect that it's been damaged, to remember what shape it's supposed to be, to stop remodeling when that shape is fixed. And so then immediately one starts to think about what might be the analogies to behavior, where the exact same things are done in the brain, right? In terms of making observations, comparing to preferences, purposeful activity, and so on. So all of these things converge on this kind of idea that we can take from neuroscience, these deep concepts, and apply them far wider than just in neurons.
Right. So what you're suggesting is our bodies are at a crude level doing a lot what our brains are doing. Our brains are just doing it in a much more specialized manner, which is information processing. But all the primitives of what brain does somewhere exist in non-neuronal cells and within our bodies as well.
Yeah, I think that's very fair. I mean, I think brains were adapted for a number of specific functions, including speed, to help in the behavioral arms race against predators and so on. But most of the fundamental things that neural networks and brains do, I think, have their roots way older. I think evolution discovered right around the time of bacterial biofilms that electrical networks are a great way to process information. It's ancient kinds of things.
So just to clarify, when you say bacterial biofilms compute or they're intelligent in some sort, what exactly do you mean by that?
Well,
in what sense are they intelligent?
Well, so intelligence is a separate issue that we can get into momentarily if you'd like. But bacterial biofilms have very brain-like, and in fact, individual bacteria have very brain-like electrical types of activity that enables them to cooperate with each other, to adapt to environmental changes, and so on. There's some older work by Koshland. There's some beautiful work from Gurul Soel, who is really tracking this kind of the propagation of electrical information in bacterial colonies, and it looks very brain-like. These are the same principles of distributed information sharing that somatic cells are doing. This is already being done in biofilms. So there's very nice new work on that.
Okay. So coming to morphogenesis, Dr. Levin, can you describe what's the traditional view of how organisms take shape and what particular we miss in that? And by traditional view, I mean the bottoms up view where DNA is seen as the sole sort of representation of what an organism will become. That's what everyone talks about, DNA as a source code of life. So what is that and why that cannot possibly be the entire story?
Sure. I think the standard view of molecular and developmental biology is that every cell has a genome. These gene products interact with each other, meaning genes turn each other on and off. So you have these gene regulatory networks that have some kind of dynamics. Some of these gene products are they're functional, meaning they're either sticky or they diffuse, or there's some sort of they're able to exert force. They do various things, and so in parallel, according to the laws of physics, all of these things interact. And then there's this emergent process whereby many cells simultaneously are doing this. And in the end, you have this complex outcome. It's the sort of idea, this emergence is the idea that, if you have lots of ants following very simple rules, eventually you get an ant hill, which is quite complex and which does things that no individual ant does. So I must say that there is nothing specifically wrong with this story. This is all true. All of these things are happening, and that's exactly what is going on if one looks at it bottom up at the molecular level. I think the limitation of this view and why I am, and others, of course, I'm not the only one who's saying this, but I am proposing a complementary, not a replacement view, but a complementary view to this is the following. In all of these feed forward kinds of processes where you have lots of very complex rules and then outcomes the rules are simple. The outcomes are complex. One of the limitations of this is that if you wanted to make a change in the system level outcome, so let's say in the case of the ants, you want an anthill that has 2 chimneys instead of 1. Then if your focus is on, at the very beginning, on the low level rules that each element is following, the question is, well, which rule do you change and how to get this complex outcome, right? So ants have rules about picking up pheromones and putting down pheromones and things like this. They don't have any rules that specifically talk about the shape of chimneys of the anthill. So what would you change? So this is known as an inverse problem, and it's in general terms, it's fairly intractable. It's an extremely difficult problem, and it leads to limitations in regenerative medicine because if what we're actually interested in is complex structure. So you can think forward that if all of the problems of stem cell biology were solved, so let's say you could make any kind of cell out of a stem cell, and the technique of genomic editing were solved, which I think it will be in fairly soon, such that you could make very precise edits to DNA. Besides the kinds of sort of single gene diseases, which admittedly are very important, then if somebody is missing an arm, or they need a new eye, or some complex outcome, or the craniofacial pattern is wrong, what genes would you edit? This is an extremely difficult problem. And so what would be nice is if there was some way of asking the question of how cellular collectives, because that's what we have during the regeneration and embryogenesis, you have this kind of swarm collective activity, how these collectives may be motivated to desirable large scale outcomes. And the thing is that I want to be clear about this. I'm certainly not suggesting that there's any sort of magic extra ingredient that somehow comes in from outside and guides gene expression. Okay, I'm not saying anything like that. For sure, at the lowest level, what you have is physics and biochemistry. That's no doubt about that. However, I think the right way to think about this is similar to what computer scientists think about when they talk about software and hardware. So what the genetics does is it gives you a specification of the hardware, right? And I think people often talk about the DNA as being the software of the cell, and I think that's incomplete. I think the better way to think about it actually is that what the DNA does is it nails down the hardware that every cell gets to have. So the DNA is a specification of low-level parts, like electrical components that you might have, all the ion channels, all the signaling proteins, everything else. That's the hardware. And so this hardware, of course, has some very robust default behavior, meaning in the absence of any manipulations, it always ends up having some particular behavior, and evolution has shaped that hardware so that default behavior is adaptive and good. And so that's how you get these bodies. But the amazing thing about this hardware is, as we've learned in computer science, you can make hardware that's reprogrammable. So what evolution has also done is given us hardware that's actually able to do many different types of modes of activity on the same hardware without rewiring. And so the question then is think about what the journey that computer science took, where in the 50s and 40s, in order to reprogram a computer, right, you had to move the wires around. You had to physically rewire it. And the reason that nowadays when you want to switch from Photoshop to Microsoft Word, you don't get out your soldering iron and start rewiring, is because we've learned that there are architectures that can be manipulated by inputs, by stimuli. And so my argument is that biology does exactly this, that evolution found this strategy. I'm certainly not saying that living things are like today's standard computers. Okay, of course they're not. There are many differences. But what's similar is the fact that there's this really interesting separation between machine and data, which means that you can alter the goal towards which these cellular collectives are working without having to rewire them and without having to solve the inverse problem and do this at a genetic level.
Yeah, so this was very surprising to me when I came across your work, the reprogrammability of biology, because I always assumed that given a particular DNA, you'll end up generating a particular phenotype or the organism. But clearly, your research suggests a lot of what we end up with is reprogrammable. So before we dive into this, I just want to make sure I understand what you're saying as correctly. So we start as a bunch of cells, which is an embryo, and due to physics, say for example, some chemicals accumulating at one end but not the other end, there is some sort of a natural structure that emerges which lets these similar undifferentiated cells into more differentiated cells. And given if you don't disturb the sort of trajectory of this differentiation, these collection of cells will end up becoming a particular organism. But what you're saying is we can intervene at some level to enable these cells to become something else entirely, even though they started as a very similar DNA structure initially.
Well, that is true. That what you said is correct, and we can talk about our particular example of that, where we make novel organisms out of perfectly standard wild-type frog cells. We can talk about that, but I want to point out one thing. This is not simply about external manipulations that cause cells to do other things. This is really a very deep inherent property of all living things. And I want to give you one example that I think illustrates what I'm talking about. And this is something we discovered in the frog a few years ago, but other people have been talking about examples of this for, even at the dawn of developmental biology. This was well understood that this happens. Imagine a tadpole. So a tadpole is a larval frog. It has very particular structure of the face and head. And in order to become a frog during morphogenesis, it has to move all those organs around. So the eyes have to move forward, the mouth has to come out, the nostrils have to move, all these things have to move. And during metamorphosis, it rearranges its face, basically. And so one could, and people did, have this view of metamorphosis that it's somehow genetically programmed. I mean, even that's a weird, claim. But people used to think that it was somehow a hardwired set of movements. Basically, every tadpole looks the same, every frog looks the same. So if you simply move if every piece of the face knows which direction and how far to move, then you get from a tadpole to a frog and everything as well. So what we did was we studied metamorphosis in so-called Picasso frogs. These are frogs where tadpoles where everything starts out in the wrong position. So the eyes are off to the side of the head, the nostrils are up here, the mouth is bad, like everything is just in the wrong place. And the amazing thing is that those tadpoles still largely become normal frogs. And the reason is because all these different things move around in abnormal, meaning unnatural paths until they get to the correct path. And actually, sometimes they overshoot a little bit and come back and have to come back. But they move around. And when do they stop? They stop when they hit the correct frog face configuration. So what you're seeing here is that this hardware is amazing. It doesn't just do the same thing because it hasn't been perturbed. It actually can start from many different configurations and get to the same final outcome. And this is this already sounds a little bit like behavior, right? Lots of animals can start from different configurations and get to where they're going because there's flexible. There's this goes back to your point about intelligence, this problem solving ability, right? Fundamentally, intelligence is problem solving in some space. So we're talking about morphospace here, the shape of all possible faces. This system is basically an error reduction mechanism. That what it's able to do, it's a homeostatic process. It can determine that there's an error. There's a delta between where it's supposed to be and where it is. And it will keep remodeling until that error is as small as it could possibly get. And of course, it's not perfect. And so you can overwhelm it and so on. But that tells you right there that what you're dealing with is a system that in some way stores or represents the path that it wants to get to. And it's able to take novel paths to that final configuration, if it can, of course, in order to do that. And so that I think is very profound. This ability of these collectives to flexibly pursue goals that don't belong to any individual cell, they belong to the collective. That I think is the key.
So I'm going to use a crude analogy, which I think captures what you're trying to say. The traditional view is probably organisms are like the top-down Soviet economy where everything is orchestrated and not top-down in the sense of having some force, but through the DNA, precise movements of where different parts need to go. But probably what you're suggesting is more like an economy where different units are trying to sort of self-organize and go to places where they make sense. Where they're supposed to be. So when you put, it's amazing you're able to make Picasso frogs. Love to know how you do that. But B, it's also amazing if you put an eye somewhere else entirely in very early stages, it's able to migrate to a place where it's supposed to be, which wouldn't be obvious if you believe that organisms only sort of develop in a certain way. So is this characterization roughly right? Of an economy versus like a top-down planning?
there's a part of that I think is absolutely right, and there's a part of that I would say differently. Let's do the top-down part first. So I think actually quite the opposite, in the sense that the standard story of developmental biology really does not like top-down control. The idea that there is some represented goal towards which the cells are working, so particularly that the system in some way knows what a correct frog face looks like or anything like that, that is not a popular idea today in developmental biology. It used to be a long time ago, but it isn't now. Even though we have many, many examples, and all developmental biologists know about these examples of systems that can get to the same outcome despite different circumstances, and then they stop. And the question is, well, why do they stop? I mean, of course, they have to have some kind of stop condition, but people usually think about this in a very bottom-up way, in the sense that we hope, and there are some models of this, but still not all that many, but we hope that in the future we will have some understanding of why tracking the low-level rules, meaning the cellular interaction rules, the biochemistry, the biophysics, molecular level rules, will somehow explain all of this, and in a purely feed-forward fashion. People are still, it is not a popular idea that the system in some way represents this goal towards which it's going. This kind of distaste for these types of models in biology stems from sort of pre-scientific ideas about teleology and things like this, and people would like to get away from that. Of course, the reality is in engineering and cybernetics, and control theory, we've had for many decades now a mature science of systems with goals, right? So it's not scary anymore. It's not magical. I think I think it's okay. But actually, that's not, these kind of top-down ideas are really not very mainstream yet in developmental biology. The other thing you said, though, is absolutely correct, which is that I think it's a useful and empirically a sort of fruitful way to look at all of these patterning systems as collections of subsystems with individual goals. So at each level, so you've got organs, tissues, cells, even subcellular organelles. All of these things are competing, even within the same embryo, within the same body. They compete with each other for metabolic resources, for information, and they cooperate. And they're trying to do various things, local, things that are achieve states that are defined at their own level. So individual cells have metabolic states that they're trying to achieve, and biochemical states. Organs have morphogenetic types of states. The cells crawl around to meet certain goals, and the final outcome is the result of all of these multi-scale kinds of activities trying to fulfill homeostatic goals that are defined at their own level. So from that perspective, it is very much a patchwork of sub-agents for what that together give rise to this amazing robustness that we see.
Right. So by homeostasis, you mean preferring a certain kind of environment or a structure. For example, a bacteria prefers a certain range of temperature or chemical gradient and so on, and it would sort of move towards an environment where evolutionarily it seems best suited. So each of the subunits we have, right from cells, organs, they have these so-called goals which are a result of evolution, right? But the question is, where is this top-down really stored? And I know this might not be even the correct word to use, because typically we use storage in the sense of DNA and so on and so forth, but these are clearly some high-level emergent phenomena.
Sure.
But still, like you said, how does the body know when to stop multiplying?
Yeah. So I think two interesting things you brought up there. Let's just we can talk about where it's stored, and we found one really interesting example of a structure that can store this information, and that's bioelectricity. There are other structures. So there are cytoskeletal structures. For example, one special thing about DNA is that it's an unbroken lineage going all the way back to our last common ancestor, right? Because each DNA is patterned or templated onto the previous DNA. Actually, there are other molecules that do this too. So the cytoskeleton is exactly the same way. Lipid structures in the membrane are that way too. And so there are other structures, but the one that we study is bioelectricity, and we study bioelectric circuits that store these kind of patterns, and we can talk about some of some of the examples that we found. But the kind of the first thing that you mentioned, can you ask that again? The first thing, there was something very interesting that you just said that I wanted to address.
I was talking about homeostasis, where
homeostasis. That's right.
It's because maybe evolution. I mean, it's an outcome evolution. That's what I was sort of just commenting upon.
So this is actually a really important point, and I just wanted to talk about that for a second. So preferences are tricky because the understanding of how to define a preference, we can homeostasis is easier to define because it's functional, and I'll define that momentarily. Preference is a little harder because it has this connotation of affect or valence. What do you actually prefer? And this is easier to visualize for more complex systems that we're used to, like animals and so on, versus simple ones. But homeostasis is easier to deal with. So homeostasis is basically a cycle in which a system exerts energy and effort to achieve a particular state of affairs, some measurable that it will actively exert energy to get back to that state of affairs. So the thermostat in everybody's home is an example of this, right? You set a set point, and this thing will exert energy. It will measure the current temperature. It will determine which direction it has to go in, and then it will act. And then it will stop acting when you land in that zone. So that's actually a pretty good analogy for a lot of what we're talking about, because the nice thing about thermostats is that you can see right away they have a set point. They have to remember the set point. Now, whether thermostats have a preference or not, that's a deeper question. But what you can see, the neat thing about these kind of homeostatic loops is that it's a very simple example of having a goal state, extremely primitive type of goal seeking system. And what's really special about it is that the goal state is encoded separately from the machine itself. In other words, if your thermostat is set to 65 degrees and you now want to set it to 80 degrees, you don't have to change the structure of the whole thing. All you have to change is the information that's stored in that set point. Leave the rest of it be, because what it knows how to do is look at that information and act on it. So that separation of information from machine is really critical. And I think, we're not so clever that we found it. Evolution found it long ago. And these set points, and we can talk about where these set points are stored. And I think bioelectricity is an important place where they're stored, much like in the brain. But that's really the cool part about this, is that it shows you a very simple way how you don't have to rewire your hardware in order to get the system to follow a different goal. And when we get to talking about the Xenobots, we can also talk about, you had mentioned that these goals are set by evolution, and most of the time they are, but it doesn't have to be that way. And I think that's actually very interesting.
On this topic of programmability, I think it's a really interesting insight that for thermostats, you could set it to any temperature and it will sort of try to get to that temperature. But of course, it's by design. We've designed it that way. I can imagine a machine that's hardwired to only seek one temperature. So in your view, why did evolution come up with this separation between the software and hardware? Why not just have everything in hardware?
Yeah, that's a great point. And I don't, I'm certainly not claiming that every biological system is that way. I'm sure there are many biological systems that are not reprogrammable, that are much more clockwork and so on. And I'm sure some of the early attempts at synthetic life, when people create these kinds of primitive cells, not the stuff that we do because we already start with cells, but really, really primitive origin of life, kinds of things. And at that point, we will see things that are not reprogrammable, at least to start with. And I'm sure evolution started that way too. I think the reason that this is so prevalent in biology is for a couple of reasons. One is I think that this architecture is incredibly suitable for challenging environments. It builds robustness. First of all, let's just think about the time scale of the individual creature, right? So you can make creatures that are sort of one-off organisms, meaning they don't heal, they don't remodel. If anything's wrong with it, it just dies. And that works. That's an okay strategy for organisms that have thousands upon thousands of offspring. And if somebody survives, that's good enough, right? And so, C. elegans, nematodes, or Drosophila those kind of organisms tend to be way more like that. But evolution already also found this other strategy where the thing's very plastic and it can actually adapt to lots of new environments. And not only is that good for the lifetime of the organism, but think about evolvability. So just the control of the system itself, never mind the environment or us trying to control it. One of the things we found some years back is that if you take a tadpole and you make an eye form on its tail, okay, those animals can see perfectly well out of those eyes. The brain has no trouble interpreting data. That those eyes can synapse on the spinal cord. They don't go to the brain. The optic nerve goes to the spinal cord. Yeah, and the data somehow ends up on the spinal cord, presumably eventually gets to the brain. And those animals can see. We can train them on visual tasks. So think about how amazing that is for evolution, because if you have this creature and things are going okay, and there's a mutation, and imagine that this mutation has a really good, a really beneficial outcome somewhere, but it also makes the eyes. It also happens to make the eyes form slightly in the wrong position. Well, if you were a sort of paint by numbers organism that had no plasticity, no good, right? Because nothing would work. All your behavioral programs would be useless. Your vision would be useless. That thing would die out. Whereas if each piece of that organism has plasticity and goal-directed activity to the point where it can actually, that eye still, even though it's in a weird location, it still knows how to put out an optic nerve and get somewhere where it can pass its data to the brain. That means that you're that much more able to accommodate new mutations. And that also means that most mutations, and any engineer knows, if you have a complex system and it works pretty well, if you start making random changes, usually things get worse. They don't get better. Yeah. And so this means that in an organism with no plasticity, it would be very slow to evolve because most changes would be harmful. Things wouldn't work as well. But if you have an organism where, if the environment of each organ primordium changes a little bit, the cells can adapt. They can still kind of do their job and get where they need to go. They have that flexibility. Then you're suddenly tolerant of many, many more mutations. They can either be neutral if the other things around you make up for it, or they can even be beneficial. But you can accumulate all of these mutations and still make an adaptive body, right? And so I think evolvability is massively potentiated by this multi-scale competency. That's kind of the name that we've given it, this multi-scale competence. The fact that at every level, cells, organs, and so on, all of these different subunits are trying to get to specific homeostatic points. And when things in their neighborhood or other things that impinge on them change, they can still get it done. And that's very important for evolution.
Yeah, I love this example so much of an eye on a tadpole's tail that's able to see. It sounds like it shouldn't be possible, but it's possible. It almost sounds like an economy again, wherein if you remove a factory from one place, everything else adjusts to accommodate lack of that factory at that place, and it could come somewhere else, and the city still functions.
I think that's very fair, because what you have here, there's a couple of related concepts that would work, in your example. One is modularity, the fact that a lot of these things function as self-contained modules, right? They can do their job in many different circumstances and many different locations. At a control level, these are basically subroutines. So one of the reasons we can make eyes, and we don't know how to micromanage the formation of an eye from all the different cell types, or a hand or anything else. But one of the advantages of this for regenerative medicine is that because these things are actual subroutines in the software of development, you can kickstart these things with very simple triggers. So very simple, low information content triggers will kickstart very complex outcomes because these are modular. And modules can be recombined, and they can be moved around, and they can be connected up in different ways, right? So I think that's really important. And the fact that all this stuff is interoperable. So they all use the same currency of communication medium. So there are conserved chemical and electrical and biomechanical signals that all of these things can use. So they can all talk to each other, even in the different configurations. And that gives it incredible plasticity.
So our bodies, in one sense, is simply perhaps the highest level of homeostatic goal. And there is lots of homeostatic goals at different levels in the body. So again, coming back to the example of, say, thermostat, I mean, homeostatic goal is simple. I mean, it's one dimensional, the temperature, but we have just so much of complexity in what our bodies and different subunits want to sort of achieve. I mean, again, I don't want to get into that preference like you were saying, but it seems very complex and multidimensional. So where is this goal state really stored? I mean, in thermostat example, you could imagine one dimensional state can be easily stored, but this seems almost too much information.
Right. So that's a great question. And I should, by the way, I should start by saying, I'm not sure even that the body is that the border of a single body is the highest level of organization, because, we have swarms and colonies and societies and so on. Who knows? I'm not even sure that that's the highest level. But you ask a great question, which is that, okay, biologists, everybody knows about feedback loops and homeostasis for scalars, for single numbers like temperature and pH and so on. How do you store the set point of a complex organism? So clearly we only have the very beginnings of an answer, but I could tell you about a simple a simple kind of set of experiments that we did to try to identify some of this. And so one of the things, for example, that we found, and this was first seen by a colleague of mine, Danny Adams, who was in my lab at the time, and she took a bioelectric map, which means just using a chemical that reveals voltage gradients. It's a fluorescent dye that you put on embryos or anything else, and the fluorescence pattern tells you where all the different voltages are. So she was observing craniofacial development, and she found that there was this thing we call the electric face. It's basically long before the actual face is made and before the genes come on that pattern the face. That sort of blank skin, that ectoderm that's going to become the face, has a pattern there that if you look at this pattern, you can sort of already decode it. It's easy to, I'm talking about that one because it's the easiest to decode. It's almost directly, it almost looks like a face. I mean, you can see where the eyes are going to be. You can see where the mouth is going to be. There's a few other things in there. So what we did was we tested the idea that this pattern was instructive, meaning that it actually controlled the development of the face. It wasn't simply a shadow or an epiphenomenon of other things that were happening. And so we very specifically altered this bioelectric pattern. And I want to be clear, we don't apply electric fields. So this is not about electrodes or putting currents on things. This is molecular physiology. So we would open and close the ion channels, which are the electrical tiny little batteries that the cells are actually using to communicate with each other, right? So we manipulated those, and what you can do is you can wipe out the normal bioelectric pattern where the eye is going to go, and you can recreate that pattern somewhere else. For example, on the gut of the tadpole. And if you recreate that pattern somewhere else, we found out that the cells are using that pattern to know what to build. And the cells will look at that pattern or detect that pattern, and that pattern apparently says, make an eye here. And it calls the subroutine of eye development, and it triggers all of the downstream stuff that is needed to make an eye. So all the traditional eye genes, PAX6, REX1, all these things that are needed to make an eye, they all turn on and you make an eye. And so you start to see that electrical distribution of different arresting potentials in that pattern was storing the pattern that was going to lead to create the face. And later on, I think one of our best examples of this, where we actually can rewrite that pattern memory, is in planaria. So in these flatworms, they have a head and a tail, and the amazing thing about these flatworms is that you can chop them into pieces, and every piece knows exactly what's missing and restores exactly the missing tissue. At the back, it'll make a tail. At the front, it'll make a head, all scaled correctly, all the right organs, and then it stops. Okay, so it has a very, basically 100% fidelity of being able to regenerate a complete worm, and then it stops when a complete worm is done. So studying that, we noticed that, and we asked a very simple question. First, we simply asked the question, how does it know how many heads to have? How does a piece know that it needs to have one head rather than two or more? And so what we found is that there's actually a very specific bioelectrical pattern that is maintained that determines where there's going to be a head and where there's going to be a tail. And we were able to alter this pattern and thus get two head worms or no head worms and various kinds of weird anatomical constructions. But the amazing thing we found is that this is a true memory in the sense that If you alter that electrical pattern, the circuit that produces that pattern keeps the new pattern. So this is a little bit like non-volatile RAM in your computer. And a lot of people hate the computer analogies because, of course, living things are not exactly like computers, and sure enough. But I think conceptually, I think this actually helps to understand it. In a piece of RAM in your computer, you can store multiple different information values, so zeros and ones and so on, in the same piece of hardware, right? So you don't need to rip out that memory or rewire it. You apply an electrical current, it shifts to a particular value, and it keeps, as long as the system is alive, it keeps that value until you come along and rewrite it. So these worms apparently work the same way. There's this circuit that has reliably, by default, it forms a pattern. This is one head, one tail, but you can rewrite it. And if you rewrite it into a pattern that says two heads, one at each end, the cells will obey that set point, and they'll make a worm with two heads, but they'll keep doing it. So if you cut pieces from that worm, you will again get two-headed planaria, even though the genetics are undisturbed. So every cell is completely wild type. The hardware is exactly the same. But what that hardware knows how to do is it knows how to run an electric circuit and use the information contents of that electric circuit to control morphology. And that circuit is amazing because it actually stores the pattern long term. So we can now shift it. We can shift it from one head to two heads. We can shift it from two to one. And we've done other work on head shape and other things. But that I think is our best example of how information can be stored that's not in the genome. And I should say, that's not the only example. Originally, people often talk about epigenetics, and nowadays they mostly mean chromatin modification, right? So these are things that control gene expression and so on. But with the original example of epigenetics from probably 60 or more years ago now was the cytoskeleton. It was found in single cells like paramecium and single cells like this. But if you cut with a little glass needle, now you imagine this, you're taking a single cell and somebody with a glass needle cutting a little square in the surface of the cell. You take that little square and you turn it around 180 degrees. And you can tell that you've done it because they have that whole surface has these little hairs that are pointed in one direction. When you turn it around, the hairs are pointed in the wrong way. So they found that all the offspring, the fission offspring of that creature, they all had the little square pointing the wrong way. And so, and it wasn't in the genome. There was nothing wrong with the genome. It was the fact that it was templating off of that cytoskeleton to build the new daughter cells each time. And once you make a change, that process remembers. It's a kind of physical memory. So there are certainly other memories than DNA. This has been known for a long time. But this, I think this bioelectrical pattern is particularly interesting.
So clearly, we've been missing a lot if we just focus on DNA as the sole storage and determiner of what organisms have to become. So, I just want to be, I just want to be clear. I'm in no way down on DNA. Okay. The science of, the science of DNA and molecular biology and all that is really important because we have to understand the hardware. And if you actually, the thing about DNA is that if you actually read the genome, what you see is protein sequence. You don't directly see anything about the length of your body and the symmetry type or any of that. You don't see any of that in there. So we all know, developmental biologists know that there's a lot more to being able to understand where anatomy comes from, right? But DNA is critical because you have to understand your hardware. If you don't understand your hardware, the rest of this is also going to be incomplete. But I just want to be on the record that I'm not suggesting we replace in any way the DNA paradigm, but I think we need to add to it in a significant way. Exactly. Yeah. So it's not obviously DNA or something else. It's DNA and something else. That's correct. Is it fair to say that as far as reprogramming bodies are concerned, we have to find like a particular level of intervention? I mean, when we naively think about biology, we think everything has to be done at the DNA level to affect the biology. But what you're suggesting is there's different level of subroutines in biology, and we just have to find the right level and trigger it to get what we ultimately want to happen.
Yeah. I think that is absolutely essential. I think in our lifetimes we are not going to have the kinds of regenerative technologies based on molecular intervention that get you what you want. The resolution to traumatic injury, birth defects, cancer, aging. In our lifetime, that's not going to be done bottom up. We have to find the most efficient level at which to control the outcome. And there are some very nice, this has been argued in philosophy for a long time, but there's actually some nice new math available where people are actually doing new information theory to show that in many systems, the most efficient level of interaction is not the lowest level. So, think about,
by efficient, what did you mean?
Well, by efficient, it means the ability to reach a desired outcome with the least effort. So we could, in theory, right, you could make any type of body structure you want by placing every atom or every cell exactly where it needs to go. Maybe someday, but that's in our lifetime, that's not going to happen. The much better strategy for us is to learn to exploit the software, the modularity, the intelligence of this collective, so that we can motivate cell groups to build whatever we want them to build and offload that computational complexity onto them, right? Not try to micromanage it. I think certainly in the near term, that's the only way we're going to get to good regenerative medicine. When I talk about the idea of levels to my students, I often give them this example. So we're here trying to reverse engineer these animals and plants and so on, figure out how they work. So you're an engineer and somebody hands you a device that's it's one of these chess playing computers, right? It just plays chess. That's what it does. But you don't know that. You have no idea what it is. So you could, of course, spend all your time focused on the silicon and the electromagnetic fields that you measure inside of this thing and the molecules of copper. And in theory, if you had unlimited time by the time, sort of the universe comes to a crashing halt, you might be able to predict what it's going to do next by following Maxwell's equations from the magnetic, electromagnetic states and following every electron as it travels down the copper. And maybe from that low level perspective, at some point, you'll make some predictions about what it's going to do. Okay. But A, that would take forever. We don't have time like that. And B, you would be none the wiser about what it was actually doing, which is playing chess. And the reason that's important, some people, people who are real reductionists, I'm not sure how many of the people are real reductionists, but a real reductionist might say, well, it doesn't matter. There's no such thing as chess. All there is electrons, moving according to the laws of physics. And I think that's deeply wrong, because if you don't know, if you miss that level, then the next time somebody shows you a wooden chessboard, which has nothing physical in common with that device, you will completely miss the fact that, hey, actually there's something very, very deep. There's a symmetry here. They have something very deeply in common, but not at the level of physics. At the level of physics, they have nothing in common. And the process of two people playing chess by mail, where they take turns scribbling things on paper and sending each other's letters, right? There's something very, very profoundly similar between that and this computer that plays chess. But at the level of physics, you will never find it. Yeah, it's not at the lowest level. So right. And so what else? What other choice do you have? Well, you might be able to, if you actually understood what this thing was doing at a higher level, then you would have these weird concepts like it wants to dominate the middle of the board, and it wants to protect the king, and it wants to get your king, and I don't know, whatever else you try to do in chess. These are concepts that don't exist at lower levels. And so if you're a traditional materialist, you say there is no such thing. But I think we can't afford to do that for regenerative medicine. I think we have to figure out what are the goal states that the system that we're given, our bodies, are able to represent. And we need to learn to manipulate those at higher levels.
Right. So on the topic of regenerative medicine, first, regenerative medicine, just as a definition, am I correct in saying that it's sort of like if we cut a finger or injure our body, how do we get to a default state all by itself?
That's certainly a part of it. A part of it is about healing from injuries and inducing, inducing the cells and tissues to natively rebuild whatever was there. And we have some great examples, right? So salamanders, the axolotl, the Mexican axolotl. That animal regenerates its limbs, its eyes, its jaws, portions of the brain. It heals spinal cord injuries. It's incredible, right? It regenerates all these throughout its lifespan. It will, if you have salamanders if you have these axolotls in the tank, they'll routinely bite each other's legs off. And in a couple of months, the leg is back and it's perfect and it works and everything's fine. they're highly regenerative. So that's what we want to get to, right? That's part of regenerative medicine, but it's actually much deeper than that, because if we had the ability to induce cellular collectives to build whatever we wanted them to build, we actually would have the solution to very many biological problems. Pretty much everything except infectious disease hangs on the idea of being able to tell cells what they're going to build. So birth defects, it's not just injury, but birth defects, meaning from mutations. And then we've shown some examples of that in our lab. Teratogens, cancer. So we can talk about cancer as disorders of morphogenesis, actually. Aging, degenerative disease, all of these kinds of things would be overcome if we had a reliable ability to tell cells what to build. And that requires us to know how do they decide what to build in the first place, and to focus on the software and on the decision-making of these agents, not just the mechanism by which they do it.
Right. It sounds like the holy grail where you're able to program bodies just like you program computers to do anything. How far have we come along there? Obviously, we've not come to, we're not nowhere near the similar state as we are in computers, but is that like a fantasy, or do you see it actually becoming a reality?
Yeah. The bad news is that living things are so much more complex than our hardware that they're harder to program. The good news is that they've had to evolve to be able to program themselves. And so they already have some very nice structures that make them easy to program. So there's kind of this balance. And so if we can discover that, I don't think it's a fantasy at all. I think it's absolutely real. How much of it we're going to see in our lifetime, I think we've already seen some important advances. I think there will be real, killer apps in this space, things like limb regeneration, tumor reprogramming, and so on. I think we will see that. I think that will be in the next couple of decades. I think we're going to see those things, but I don't have a crystal ball, so I can't tell you how the research is going to go. But I don't think it's a fantasy, and I think long term, when I give talks, one of the first slides is I say, what are we trying to do here? What's the end game? When can we all go home and say we're done? And the first thing that I show is what I call an anatomical compiler. So I think we need to get to the point where you can sit down and you can draw using a CAD-like tool, right? Like if we draw, car parts and things. You should be able to sit down and you should be able to draw the kind of animal or plant that you want at the anatomical level. And much like with computers, where you draw these things and you don't worry about where the atoms of each thing are going to go and what you specify at the large scale, and then the compiler can give you the low level events that need to happen for this to take place. The same thing. So you should be able to say, okay, I want something that's kind of like a frog, but I want it to have 6 legs, and I'd like it to have a head that looks like this and a tail and whatever else. And once we know what we're doing, the system should be able to go from that description to a set of stimuli that it would give to cells to cause them to undergo morphogenesis for that. That's our dream. And are we going to get to see the full sort of general version of that? Probably not, but specific examples of that, I think, are absolutely coming. Yeah.
Right. And this programmer or a compiler will not just give us ability to heal, but it will also give us the ability to create living machines.
Yes.
I know you've been working on things called Xenobots, which are V.01 of what you are describing. So can you tell more about what are they and how do you see them sort of evolving from here?
Sure. Yeah. The Xenobots, and the reason they're called Xenobots is because the initial ones that we made were made from cells from the frog called Xenopus laevis, and they're biobots, so we call them Xenobots. It was really an effort, and this is done in collaboration with our colleagues at the University of Vermont, Josh Bongard, in his lab. And the important thing about these Xenobots is that they're a first example of a much bigger emerging field that are basically synthetic organisms. And the idea is this is different from synthetic biology, where you're moving DNA between different bacteria and things like this. This is really designing new bodies along with new cognitive capacities that these different bodies are going to have. And the idea is, sure, we'll make some useful synthetic living machines, and they'll do things like clean up the environment and chase down cancer cells in your body and, scrape plaque off your arteries and things like this. But the deeper issue is that if we understood morphogenesis well enough to make bespoke organisms designed to a particular anatomical and behavioral spec, then all the problems of regenerative medicine would be solved, and you could build and improve biological structures at will. And so for the Xenobots, now one simple question there was even, is there even any plasticity? I mean, people talk often about developmental constraints, meaning that, hey, look, you're a zebrafish cell. What could you make but a zebrafish, right? That's the kind of the standard assumption. And so, and we even before the Xenobots, we've had some interesting data on making planarians with head shapes that belong to different species of planaria without touching the genome, basically accessing different regions of that amorphous space bioelectrically. But we just asked a simple question. We said, okay, if we take some cells, in particular some skin cells from a frog, an early frog larva, we take some of those skin cells and we let them reboot their multicellularity. We let them sort of get together in a different environment away from the rest. We liberate them from the rest of the animal. We say, okay, you're on your own. There's a group of you. What do you want to do? And there's a bunch of possibilities, right? So one possibility is that they just die because they're single cells and they don't go on. There's a scenario where, well, maybe they just make a flat monolayer where they live in cell culture, every cell for itself is reproducing as fast as it can. Maybe it might make a tumor. There's all kinds of possibility. What they actually do is something much more interesting than that. They really do undergo multicellularity in the sense that they coalesce, they combine, they come together, they signal to each other, and they form a new creature. And this new creature, why do I call it a creature? It has a very defined anatomy. It has behavior, so it has the ability to move around. It has various behavioral repertoires. It can do certain things. And the cool thing about this creature is that it doesn't look like a tadpole. It doesn't look like a frog. It doesn't look like anything you really would have predicted. It has a standard frog genome. So one way I often give talks about these things is I show 10 or 20 slides of videos of these things running around and doing various things and changing their shape and healing from damage and interacting with each other and interacting with things in their environment. And they say, what do you think this is? And people say, well, you found it in a pond somewhere. It's some sort of some sort of underwater creature. And I said, well, we have the genome. And, the genome. Well, that's great. Let's sequence the genome and we'll see what we learn. Well, the genome was 100% frog. It's 100% Xenobots. So this tells you right away a couple of interesting things. It tells you that cells have incredible plasticity, that they like working together to build things, but it doesn't necessarily have to be the default thing that they normally build. So in novel environments, they can build other things that are coherent, adaptive structures. So these things these Xenobots, they don't look like a mess. They look like a creature that might exist. It's just a completely different body plan, but it's perfectly serviceable for what they do, which is getting around and so on. So that's one thing. The other interesting thing to think about is that usually when you're looking at some sort of organism and somebody asks the question, okay, why does this thing see green light, or why does it have wings, or why does it jump, or whatever it does, the answer is always the same. Well, because for millions of years, its ancestors were rewarded for this or that. And so it has this long selection history towards a very specific outcome. Okay. The cool thing about these Xenobots, and these Xenobots are just the first steps of a field of this new field of computer designed organisms that are going to be new model systems beyond the standard, zebrafish, frog, mouse, whatever that people have been working with. There's an incredible option space for new types of bodies. And the thing about these Xenobots is that they don't have an evolutionary selection history back on Earth. They were created. They create themselves in 48 hours in front of your eyes. And now the individual cells, of course, do have a selection history as being part of frogs, but they were selected for the property of sitting quietly on the outside of a frog and keeping out the bacteria and fungus. They were not selected to have this amazing property of running around and doing various things. So that shows you this incredible plasticity, and it shows you that we need a better way of understanding and predicting goals and capabilities when there isn't a neat evolutionary backstory about where everything comes from, because it can arise de novo from scratch. And so that leaves us with a very interesting question of where do they actually come from? So the body plan information, certainly it's people are often kind of surprised about the fact that you can't read this out of the genome, right? When you read the genome, there's just frog. You can't read this new information. However, what's often not talked about is that you actually can't read out the original frog shape from the genome either. We have no idea how to do that. What we can do is compare that genome to other animals that we do know what they look like, but that's cheating. What we really want to do is be able to look at something at the mechanistic level and say, okay, I can figure out what shape this is going to be, and we don't have that ability yet. So, I think these Xenobots, in addition to potentially being useful living machines, they give us the ability to really, they force us, in fact, to really reconsider our some deep questions about where body plans come from, and how physics, information, and genetics all interact to determine what's possible for a living organism.
Right. So could you change what these Xenobots shapes end up becoming, or do they just self-assemble into specific shapes?
Yeah, we're working on that now. So the two papers that have come out, one last year and one just came out this Tuesday, I think. In those papers, we are just characterizing what these things do naturally, because I think it's really important. So it's funny. We made the choice to focus initially on what they do completely naturally with very little manipulation from the outside. We could throw in new genes, and we can throw in nanomaterials and do all kinds of stimulation, and we will, and we're doing all that. But as, when you do that, people will say, well, that's a fine example of bioengineering, and you've made them do something very unnatural, and okay, that's great. That's synthetic living machines. But people will say, but that has nothing, that doesn't mimic in vivo, and that doesn't have anything to do with evolution, whatever. Okay. So we wanted to start with a much more foundational point. There's been other biobots, not of this type, but there have been other biobots in the field as examples of bioengineering, precisely bottom-up controlled biobots. What I wanted to do here was say, this is native. This is endogenous plasticity. Look at all the things these guys are already doing before we even really did much to them at all. And especially the second paper. The first one had a little bit of sculpting, and it guided by the computer evolution, which is cool because in that case, their evolutionary history didn't happen on Earth. It happened in a virtual world in the Bongard Labs, virtual environment. All the evolution took place there. They literally evolved these things. And then we made them, physically real. In the second paper that just came out, there wasn't even that degree of manipulation. It's completely emergent. And I think it was very important to me to nail that down first, so that people can see this as spontaneous, meaning natural plasticity, not just a weird trick of bioengineering. But the next step, of course, is programming them. So the next step is going to be, what kind of stimuli can we give these cells? Maybe they'll need new synthetic biology circuits in DNA. Maybe they won't. We don't know. We need to figure out what preferences they have. We need to figure out what learning they can do, simple or complex or what. And then we're absolutely going to start programming the shape. Yeah.
Right. I mean, it just seems like you say it's early stages, but you could very well imagine a couple of decades or maybe more ahead where you are programming a much more complex organism. And you say, I want this, and there is some automated machine that ends up recreating that organism. And particularly your comment on if evolution on computer is just mind-blowing, because they just don't share the natural history, and we can make them do whatever we want to.
Yeah, absolutely. And we're already working on that. We're working on automating this process so that it's both high throughput and directly linked to the digital evolution that happens, in silico. And my interest is to open up this incredible option space of new bodies that are combination. I mean, already, right? So some of this already exists. We have people with all kinds of smart implants and people who drive wheelchairs and other things with they're directly with their mind. So they're directly hooked into physical devices. And we have other robots that are instrumentized with living cells on them and things like this. So this merger of biology and technology is kind of starting. But I think we really need to expand the model systems that we have beyond standard, zebrafish, frog, and axolotl and Drosophila, whatever. There's this incredible option space of new animals and plants that no one has ever seen before. And we need theories that can handle that. They're not just tied to specific model systems.
Right. I think we want something of an equivalent of like a Fortran, Pascal, or BASIC to start like programming these systems, the anatomical compiler that you're suggesting.
Yeah, that's true, and I think it'll go in both directions in the sense that once we understand how the biological software works, I think one of the coolest things we'll be able to do is actually copy that or adapt it for machine learning, for robotics, for swarm robotics, for various kind of engineered artifacts. Because I think there's lots here that it's not just from the computer science to the regenerative medicine, which is a lot of what we've been talking about here. It also goes the other way around. So I've given a talk called Why Robots Don't Get Cancer. And the idea is to really think about this multi-scale competency as a new architecture for robots and for machine learning that really builds on how biology does things.
Okay. So Dr. Levin, I wanted to ask something that must be sort of asked you multiple times, but I'm going to ask anyway. It's about ethics. How do you handle ethical dilemmas, which inevitably crop up when you're working with intelligent and maybe likely conscious multicellular organisms? How do you make that trade-off?
Yeah, that's a very important question, and I think the thing to really keep in mind is that it is completely unconscionable to not do research that ends up helping in medical outcomes. So I get phone calls every week from people with kids with birth defects, with accidents, spinal cord damage, amputated limbs, whatever, you name it. I get these phone calls. People are absolutely desperate for help, and their question to me is always number one: hurry up. They say, what's taking you so long? I'm reading papers about frogs and worms. Yeah, that's fine. When can I come in and get my kids healed? And so what you really have to think about is the opportunity cost of not doing this kind of work or of slowing down this kind of work. I've heard it before that, oh man, this is scary. Maybe you shouldn't do it. Okay, that all comes from healthy young people who don't know what children's hospitals are for, and why medicine exists. It's I think it's a morally untenable view to slow down experiments with frog skin and things like this, if it's going to slow down the relief of suffering. I don't have stats, but you can imagine how many people, if you add up cancer and birth defects and degenerative disease and everything else, we need to be working on this as fast as we possibly can. Not to mention, so that's the biomedical side. The other side of this is that I actually think it's an existential risk for society as a whole to fail to understand how goals and preferences arise. Because we, for sure, in our lifetime, we are going to be, we kind of already are, but even more so, we are going to be inundated with swarms of all kinds. Software swarms like the internet, Internet of Things, swarm robotics. This stuff is not going anywhere. There's no avoiding it. It's going to be here. We do not have a mature science of tracking where goals come from in swarm agents. We have no idea. So you put something together, maybe you understand the individual pieces, but the collective, what is it going to want? What are going to be the homeostatic states that it's going to exert energy to achieve? We don't have any idea. And if we don't know how to control it and predict it, we are in big trouble. And I think these kinds of experiments are a sandbox in which to take very sophisticated agents, these cells, and ask, fine, can we learn how to predict and then control what the collective is going to try to do? So I think human flourishing absolutely requires science. I think there's no way around that. We don't have any other way of improving the welfare of everybody. And if you want regenerative medicine and you want to survive the coming technology as a society, to survive the technological changes that are coming, this kind of work, I don't just mean my work, I mean all this, the field, the community, the things that everybody's doing are essential. It is simply unconscionable to slow this down.
I agree. Whenever we talk about ethics, I think we always look at the one side, but there's always the other side, in the sense of who is it going to benefit? but we always look at the scary parts, and media doesn't help when it just focuses on the isolated incidents and never mentions all the benefits that come out of such research.
Absolutely. And I mean, of course, there's oversight and everything we do, even with frogs, there's a million permits that we need to get as far as ethics boards and whatever. So there's already an existing structure that oversees all of this kind of stuff, including experiments that other people do with mice and so on. But the thing is, people often, the calculation that people often do in their heads is this, well, everything's great now. And then if we do something bad and scary, it'll make things worse. So let's not do that. But that's a completely false dichotomy, because things are not great now. We have massive unmet needs, and not taking efforts to make things better is as bad as making things worse. We don't live in this paradise where we just need to make sure we don't screw it up. That's not the world we live in. We have massive disparities in healthcare, both economically and in terms of quality of life. And this needs to be addressed. We can't afford to just sit there and hope we don't mess things up. We're not in that good a shape.
Right. So, Dr. Levin, I wanted to spend last bit of our conversation around scientific creativity. I mean, as a meta topic, you have your lab and your group and your research is just amazing source of so many great insights. It's almost seemed like, it's a bedrock of how biology will evolve from here on. How do you, I mean, I know it's very broad, but how do you get or enable this sort of creativity within yourself and amongst the students that you work with? And is it a teachable skill?
Yeah, interesting question. Well, I think it is a teachable skill. I think that, like any other skill, people come to it with different baseline levels of talent, and then from there you can sort of improve. I think it is teachable. For myself, I use a variety of techniques. One thing that I do is I sort of read pretty widely. So I try to make sure that I'm looking at problems from multiple perspectives. I mean, I think that's lots of people know about that. Where I ask, how does this certain problem look like from the perspective of a physicist, a computer scientist, a biologist? I try not to stick. And it's funny when I give a talk about these things, so I have an hour-long talk that I give to professional audiences. I always know what kind of department I'm in based on which part of the talk makes people angry. And it's a different part of the talk every time, because certain things you can say in one department are completely obvious. And if you say those things in another department, people throw tomatoes because it sounds ridiculous to them. And these are departments that might work on the same floor of a research institution. And so it's clear that we're very siloed still, in the sense of conceptually, right? Which things are ways to think about problems and things that are considered weird versus obvious. So what do? I try to read widely so that I have different perspectives on the problem. Another thing I try to do is I really try to have a cyclical process of alternating sort of unconstrained creativity and logic and hard inference, so that you have periods where you're working or thinking about a problem where you say, okay, during this time, I'm going to really get wild and kind of think as far outside the box. I'm going to question all my assumptions. I'm going to take things that we pretty much assume, and I'm going to say, okay, if that's wrong, well, what then? What are the implications, right? If assuming that that's actually completely wrong, what does it mean? And so you do these kind of creativity exercises, and then you get some ideas. Now it's time for the rigor, and then you test them very very rigorously. And it's okay. Is there any value to this actually, or was it nonsense? And so you go through a period of that, and then you get some results, and you learn some things. You fail a lot. Maybe you have a success somewhere, and then you go back around and you say, fine, now knowing what I know now, let's do the creative phase again, and let's just brainstorm about different ways to look at this, and how can I take things that I've heard in other fields, and I can like one very important tool that I use is I look for symmetries, and I look for my early work was in left-right asymmetry. I love symmetry. I always look for symmetries and things, isomorphisms. So can I take things that exist in one field and map them onto my field? And some things will map and some things won't. And then I take things, and then I look at the things that don't map, and I say, what might be there that we just haven't found yet, right? The gaps, now the symmetry might be bad, and then you throw it away eventually. But if it's good, the places where it doesn't map, that's a pointer to things you haven't found yet, because maybe there is something there. I give you a very simple example. It's an exercise that I've done with some of my students. I say, take a paper in the neuroscience field, right? A paper on the brain in neuroscience, import it into Microsoft Word, and do a find and replace function. And you take every time it says neuron, you replace that with cell, and every time it says milliseconds, you replace that with hour. And then you read the paper and see if you can learn anything about developmental bioelectricity from that. And so then what happens is you read these things and you say, oh my God, of course we have this in development, and we have that too. We have this other thing that's going on here. Yeah, of course we have that. And so these kind of symmetries, right? Making that pivot. That's a favorite technique, is to take and sometimes it doesn't work. If the analogy is bad, it just, then it's bad. Nothing comes of it. But we've had really good luck with that kind of thing. And that's what I try to teach my students. That's the kind of thing.
So you're importing these patterns of thoughts and mental models from various fields, from neuroscience, from physics, from computer science, from machine learning. But I think people who specialize in one field, and they sometimes argue that it's hard to read so widely. So because there's just infinite complexity in any field that you may want to read about. So is there sort of like a tactic or a strategy or a thumb rule you use that this is enough? Or because computer science in itself could be infinitely large, the literature there.
Yeah. Everything is an infinite literature, and I have a massive stack of papers I haven't gotten to yet. Of course, there's never. I don't have any magic. I don't have any way of making that manageable. Inevitably, the challenge is bigger than any of us can practically muster. However, just because you can't do everything doesn't mean you can't do something. So you can still make progress. And one other sort of tool that I teach my students is to really hone your intuition, and it takes practice, about what's essential and what's not. So if you get we very clearly see these examples. So I have a lot of computation in my group. We have all kinds of people. We have biologists, we have computational, programmers, whatever. And you can see this right away when you hand a biology paper to the computer scientist. The computer scientists are actually very good at this. They're very good at compartmentalizing and coarse graining things in a logical way, and trying to figure out, okay, what are the details here that matter and don't matter. And so my job, I'm sort of the kind of transformer that sits between all these different disciplines in the group, and I try to kind of make sure that they're all talking to each other. When I give someone a paper, I will help them. I will say, look, this is a 25-page paper. You don't need 90% of the information that's in here. You don't need that. Here's what this paper is saying that you need to dig into and you need to understand. So to the extent that, and of course, sometimes I'm wrong and I miss things. I mean, that's just, this is never going to be perfect. But the goal is to go through all this and not get bogged down into the details, and to really, that makes the literature a lot more manageable, because you ask yourself, for any given thing you're trying to read, what actually is the core here that's important? What am I going to import from this that I can use somewhere else? And what are just irrelevant details that are never going to matter? And if you can get good at doing that, then it makes things a lot more manageable. But I'm not going to pretend there's some magic where you can use all of the insight of any of these fields. You bite off as much as you can. That's all you can do.
Right. This is great, Dr. Levin. I mean, it's been great chatting with you. I hope that you and your group are able to accelerate towards this universal and living creature compiler, so that we heal all sorts of diseases. It just seems, talking to you and understanding all the research, that this seems a very pivotal moment in history. And I wish we are able to realize all the vision you have in reality.
Well, thank you very much. I appreciate that. I think you're right. I think this is an incredibly interesting time. I'm super pleased to be part of it, to be able to work with the many people. I mean, you and I have talked for an hour, but this is the stuff that we've been talking about is the product of the hard work of lots of people, not of course outside, but also in my group, postdocs and students and everybody else. It's a huge kind of group effort in society, and I'm super pleased to be part of it. I think we're going to see amazing things in the next couple of decades. We're going to see amazing.
Indeed. Thank you once again for your time, and all the best for your research.
Very good. Thank you so much. I appreciate it.
Have a great day. Bye-bye.
You too.